Everyone, very excited to be part of this 2 days webinar. Also very grateful for this opportunity to share my research on rhododendron and its association with climate and pollinators, with all the researchers and the rhododendron enthusiasts in general. So today I will be giving you an overview of my 3 studies, which I carried out for past few years for my PhD and my postdoc. I'll be starting with a study which was carried out in the Sikkim Himalaya and which forms one of my key chapter. There I've studied the phenological events in Sikkim. Second, I will be presenting you the variation in floral traits and plant-pollinator interactions between the North American native rhododendron species and Himalayan species. And I'll be ending the talk with some of the preliminary findings of my work on macroevolution of some of the reproductive continuous variables. So let's begin. So having grown up in the mountains, I feel personally very connected to the mountains, and I've been working in this landscape for the past almost 10 years now, and I find mountains very unique. Of course, it's also known as the natural laboratories, mainly because of the natural environmental gradient it provides as we move from the lower to higher altitude. Now, because of these environmental gradients, we see the changes in species distribution, composition, phenology, and functional traits. Now, understanding this dynamic is important. Also, generating information is important, even to make some general prediction about how global climate change may have an impact on the vegetation, both flora and fauna, and also to maintain a long-term ecosystem stability. Now, this natural environmental gradient not just have an impact on the flora and fauna distribution and composition, it also has an impact on its interactions and which is plant-animal interactions. And I'm specifically interested in the mutualistic interaction between the plants and pollinators. Now, let's understand why is it important to study this amazing mutualistic interaction between the plants and the pollinators. So we have many amazing work carried out to show how the pollination process is extremely important, or it is one of the contributing factor for the diversification of flowering plants. And so what happens is the floral visitors, which efficiently pollinate, and so which are pollinators, select for specific floral traits. Now these traits over a period of time becomes dominant in the population. Now this is one of the, or this idea in general is the key concept of pollination syndrome hypothesis. Now I'll explain this further with the help of the traits, both in terms of plants and the pollinators. Now, when we talk of plants, according to the pollination syndrome hypothesis, we see that plants are attracted in case if they are pollinated by birds. So the plants often has a flower which is red in color, or usually warm shades, I would say, and are either tubular, narrowly tubular, or narrowly campanulate in shape. And in terms of nectar, those plants prefer nectars with high voluminous nectar volume and less concentrated. Now, again, the phenology events, which is again the key reproductive traits in the plant, is also seen to vary. For example, the staggering or overlap in the phenological event has also been associated with the competition within the pollinators, and also when the pollinators actually facilitate the pollination process. Now, coming to the traits in terms of pollinators, so it's often their preference for the reward. By reward, I mean either the nectar or the pollen. It's determined by its body shape, how much energy they would want, and its mouth shape or beak, and also the food preferences. Now, in today's time, where there is important role which is being played by the climate on the plant-pollinator interaction, it becomes important to include climate as well when we study the floral trait evolution in plants. And again, apart in addition to the climate, it again becomes important to consider the evolutionary history of the plants which we are studying. Now, in this context, I chose rhododendron as my study system, and here I would share with you some insights on its association with the climate and the pollinators. Now, why did I choose rhododendron for my work? So, or in general, why would you study rhododendron to understand the floral trait evolution? So, of course, one major reason would be its amazingly distinct ecology, its geographical distribution, both along the elevation and its altitudinal, latitudinal and longitudinal gradients. Second would be its variation in the reproductive traits, starting from phenology to the floral morphology, and also, so these would be in general the ecological importance, which is very evident and beautiful to see as well in terms of rhododendrons. But when you talk of rhododendrons, especially which are native to the Asian mountaintops, the importance of rhododendrons becomes much more than just the ecological importance. So rhododendrons here serves a major ecosystem service. So I've here taken the example of Basnett Rhododendron Sanctuary, which is located in Sikkim. So the flowering attracts a lot of tourists, of visitors in general, which can be photographers, trekkers, researchers, or in general public. So this is the timing where there are a lot of small entrepreneurs would earn for their living. And so yeah, that's how the people get benefited during the peak flowering timing, which stays for almost 3 to 4 months. Now, some of the rhododendrons, such as rhododendron arboreums, it's edible. So that's also used for the preparation of wine, both in the eastern and the western Himalayas. And they are sold locally. So again, people benefit out of it. Now, apart from that, especially the high altitude species, which has a very high fragrance, they are used for making incense. Both flowers, leaves, as well as the stems is used for making incense. And using of incense early in the morning is especially very or it's considered to be very important, especially in the northeastern region of the Himalayas and also many parts of the Asian mountains. Coming to the providing habitat for the wildlife. Now, this characteristic of the rhododendron forest, I realized this only when I started doing my field work and started spending more time in the forest. Now, here are the images from my field site in Kengusla Alpine Sanctuary. So what I observed during the field study is it's supporting a lot of wildlife habitat. For example, here is a musk deer, which is the endangered species of the region. So what happens is during the winters, when there is absolutely no resource, rhododendron being the only woody flowering plants, and the species are mostly evergreen. So musk deer feeds on the rhododendron leaves during the winters. And rhododendron provides a very good thickets. So it co-occurs with the conifers and provides a very good understory and thickets for the beautiful pheasants in the regions, such as the blood pheasants and the Himalayan monal. Now, apart from that, even from the climate change perspective, I see rhododendron as a model system to understand how the climate has an impact on various phenological events. So studying of phenology is also known as a bioindicator for climate change, and especially for the regions where we don't have long-term plots, studying phenology across the elevation gives you a very good understanding about how the climate has influenced the phenological events, and or just to have an understanding, is phenological events influenced by or associated with any kind of climatic changes. Finding these kind of findings would be very useful for the future researchers. So so having all these importance for the rhododendron, I carried out the local scale field study. Now, this is one of the PhD chapter I did in India, here, followed by the regional study where I got this opportunity to do my first postdoc in the North America, and that's where I did my comparative study on the plant-pollinator interactions. And currently, I'm focusing more on the macroevolution of some of the floral traits for the particular genus, so subgenus. So I'll start with the local scale work. And here I ask two questions. Can abiotic factors and evolutionary history explain the phenological events in rhododendron species? And is there a relationship in flowering overlap and pollinator and phylogenetic structure of co-occurring rhododendron species? Now, as I mentioned, the study was carried out in Kengusla. So I'm using this image just to show the elevation gradient, although I covered the elevation gradient from 3,400 to 40 to almost 4,200. The gradient here just show the elevation just below the tree line, and the red dots are the plots that was laid across the elevation. Now, when I decided to do my field work in Kengusla, which actually provides a very good elevation gradient without having to trek much, which is usually the case in most of the Himalayan mountains, this was almost like a dark spot where I did not have any information of what all the species are and how the species composition would change across the elevation. So I started off by doing very conventional way of understanding what the vegetation looks like. And that's when I decided to lay plots, which is of 20 meter into 5 meter. So 27 such plots were laid across the elevation. Now, this was important information for me, because this would then let me understand what are the species that I need to select for my phenology monitoring. And then I carried out the phenology monitoring for 15 days, at every 15 days interval, and my sampling was for 3 years. So I continued till 3 years, because the region like Himalaya, it's so uncertain. Usually the first year study doesn't make any sense, to be frank. So it took me just to understand how the elevation gradient, elevation gradient influence various ecological events or even the functional traits. So yeah, especially for the phenology, I decided to have some good temporal scale information. So then I wanted to have an understanding about the role of the abiotic factors, and for that I installed eye buttons at every, at certain intervals across the elevation, and eye buttons were installed both for the atmospheric temperature as well as the soil temperature. And for the weather station, I was part of a big project. So they had installed the weather stations already around some of these plots. So that was again helpful. And day length, I used it from the geosphere package. And so the larger tree was built, phylogenetic tree was built, and then I pruned the tree for the species of my interest. So I used various phylogenetic comparative methods for the analysis and the model selection, of course, with the lowest AIC values. Now, this image I wanted to share you just to show how the gradient looks like. So I'm actually with my field assistant right after the laying some of the plots, and we are at almost almost near to the tree line, 3750. And so these are the regions above the mountain tops, as indicated by those arrows, are actually above 4,000 meters. And then those areas had the last plots for the vegetation as well as for the plant pollinator interactions. Okay, so coming to the first question. So what I did was I selected species and then looked for the events. So I studied the budding, flowering, initial fruiting, immature fruiting, and mature fruiting. So I further for my talk, I'll be referring the first three events as early events and the last three as the late events. So again, here's a picture just to show you how the vegetation or the phenology changes in the landscape. So actually this particular area is exactly the top, the arrow, with the arrow what I showed you. So this is how the top area looks like, the mountain tops. So we have a lot of glacial lakes, and that makes the region looks even more beautiful. So the three image, firstly, the mid-flowering, mid-June, where the flowering happens for rhododendron erosionism, and early September, where it sets fruit, and end of October, where it starts dispersing, where it sets open the fruit's dehiscence. Now, so in the plots, I came across actually 12 species inside the plots, and this kind of composition or the number of species has also been observed in the work carried out in China or across almost similar elevation. So, but I couldn't include all the species which were inside the plots because my overall motive was to understand the role of elevation on phenological events, and for that I needed a lot of replication across the elevation. But these species were rare, and it was very difficult to find individuals for replication, and again for the pollinator studies and my functional traits measurements. So that's how I then later decided to work just on these 10 species. Now, moving on. So what we found was there's a strong phylogenetic signal observed in early events. So what it means is, so among the stem species, the early events, budding, flowering, fruiting, happened during the similar time. So even if the individuals were distributed across the mountain, it would still show these phenological events or share the timing of these phenological events. Which was quite interesting. And here is a phylogram to show you how it looks like. So the color gradient here is the day of a year, or you can just refer it as the different months. So you would see the Thompsoni and you'd see the first three species which buds during the early May, followed by the other three, and the last three species, which are usually the high altitude species, which buds during the mid-June and end of July. So I did not see this signal for the late events, but yes, the early events were seem to be conserved across the 10 species. Now, when I looked for the role of the abiotic factors, what it showed was the early events were, or the model which explained the most variation in this early event had these two variables, day length and day temperature. So why I say day temperature is, again, the night temperature did not show any significant result, and I did not include the soil temperature because then soil temperature was highly correlated with the day temperature. And the reason for taking soil temperature is because, of course, this snow melt is very important, but then it was very expensive for me to have anything or any instrument to measure the snow. But I mean, I've seen many studies use the soil temperature as a surrogate for the snow melt timing. So yeah, that's how these two variables seem to be important. And also I found a significant role of the evolutionary history, which is explained by the Peegel's lambda. Whereas the later events were not not influenced by the day temperature. The model just had the day length, and day length seemed to be an important factor influencing the late events. Okay, so again, further for my second questions, what I did was, so at every 100 meter, I could see that the species composition would change. So what I did was I carried out pollinator visitation study per species at every elevation. So, and then I had multiple replications just to have enough sampling points, and then calculated the pollinator similarity index, and then flowering phenology overlap index. Again, this was calculated for every elevation belt. So altogether, I had 9 belts, and the commutative phylogenetic structure was also calculated for each of these belts. So coming to the result, so in my y-axis, I have the flowering overlap index, which is my response, and my predictors are net relatedness index and pollinator similarity index, and the color gradient here shows elevation. So what I found was the pollinator similarity index decreased in the lower elevation and had a negative relationship with the flowering overlap index, whereas net relatedness index showed a positive relationship with the flowering overlap. So in the lower elevation, there was more overlap in the flowering timing. Here is an example of the low elevation, how it looks like in the Eastern Himalaya during the peak flowering timing. So just to show you how the overlap would look like in the region. So I would want to explain this further with more illustration. So I'm referring to here low elevation because I'm considering all those belts below the tree line. What I found was the birds were the important pollinators, and of course I had nectarivorous and non-nectarivorous birds, and various diversity of bird visited the species. Bumblebees followed by the other bees were also seen as the common pollinators, but bumblebees would always number would always be more, followed by flies. So I observed less pollinator similarity. Here I am referring to the abundance or the each visits that I looked at, and the species, rhododendron species studied in each of these belts were closely related in each of these belts and had more flowering overlap. While in the higher elevation, so bird visits significantly decreased, and visitation made by the bumblebees increase, and again the flies also seem to have prefer more high altitude species. And more pollinator similarity. They were, however, more distantly related in the higher elevation, the rhododendron species, and I found less flowering overlap. less flowering overlap in the high altitude species, co-occurring species. So what I would conclude is the closely related species share the timing of early events, including budding, flowering, fruiting, and initial fruiting. And my results show that the later events were strongly associated with day length and not constrained by evolutionary history. And lower elevation species were closely related, shared high flowering overlap, and lower pollinator similarity. Now, all of these work has already been published, and if you wish to know more, you can always refer to the following papers. Now, moving on to the regional scale study, here I asked two questions. Can the identity of pollinator explain floral trait variation in genus Rhododendron, and are climatic condition associated with floral traits and pollinator identities? So this work was carried out in Sikkim, and which is a small state in the Himalaya and also in North America. So in Sikkim, I expanded my work more to the lower elevation because in Kengusla, I found that birds are more in the lower elevation. So I again imagined that if I go below 3,000, I would see more birds. And so that's why I selected Percy Rhododendron Sanctuary and another location in Lachen Valley. Again, for my first postdoc, I got this opportunity to work in the US. And of course, that's how I studied native North American rhododendrons in North Carolina and Maryland. My focus was mainly to cover the Appalachians. But I did study a few individuals outside Appalachians as well. So altogether in the Himalaya, I sampled 17 species, and in Appalachians, it was 7 species. And here are the species which are the flowers of the species which I studied altogether. Okay, for this work, my response were floral traits, the corolla length, which I measured in the field using the vernier caliper, and nectar. So nectar concentration and volume were measured. So how you do that is a day prior, you cover the flowers with a net just to avoid any pollinators visits. And next day you would go and during the peak pollinator visit timing, you would open the bag and then measure the concentration with refractometer and use micropipettes to measure the volume. And here my predictors are the pollinators, visitation, and I just did the day visitation, and that's what it is. And based on the result, I grouped them into 4 different groups, which is the bird, bees, flies, and butterfly. And again, pollinator visitation study was carried out for all species, and there were multiple replications done. And just considering the species that were included, I used Ding et al. Rhododendron phylogeny and pruned the tree for species of my interest. And for occurrence point, I used GBIF and of course the field data as well. Now, I would start by giving you just an idea of how the trait variation looked like across the 2 regions. What I found was, in terms of nectar concentration, overall, it showed a significant variation across all species, but it was very interesting to see that nectar concentration was much higher among the Appalachian species, which range from 13 to 33 percentage, while in the Himalayas, it was from 3 to 28 percentage. And here are some of the species from the Appalachians, which showed very high nectar concentration: Calendulicium and Rhododendron maximum, and Ceticism from the Himalayas. Now, when I looked at the nectar volume, I had a completely opposite trend. The North American species showed low nectar volume from 2 to 8 microliter, whereas there was extremely high nectar volume among the Himalayan species from 2 to 81 microliter. Now, here are some of the images of Himalayan species which had which had the highest nectar volume, and they are often the warm color species. And I also measured the corolla length. Overall, we found significant variation across species, but more variation among species was observed among the Himalayan species, which is quite obvious given the higher diversity in the region. But something which I have not worked on here is the higher floral shape in the Himalayan species, and which is worth investing more. So the Appalachian species, it range from 32 to 42 and Himalayan species from 13 to 59 Then, so what I did was I wanted to understand the pollination syndrome in the genus. So I considered all the species and as a group, and then tried to see if the pollinator explained floral trait variation in the genus. What I found was nectar concentration was negatively associated with a bird and positively associated by the bee visit. Now, for the analysis, I've still considered the outliers you see on the screen. Even if you remove them, you would still see a very significant trend. But I would want to show these points because this is how the variation looks like within the species, right? So still, the nectar concentration was negatively explained by birds and positively by bees. Now, even while doing the analysis by considering just the Appalachian species and then next model for the Himalayan species, I would still see this trend clearly. Okay, so in the Appalachians, I saw bumblebees to be extremely active, especially for the species which belong to the subgenus Haemonanthus, such as Catawbancy. In fact, that amount of bumblebees I've never seen in the Himalayas, and my reading would just shoot up. So it was quite busy pollinator activity observations. And in the Himalayas, we could see bumblebee only in the higher belt. But what was interesting, although my trend was not very significant, is nectar concentration in the Appalachians is also related to the butterfly. Usually, the flowers with extended, loosely bundled, upward reflect stamens and pistils seem to be efficiently pollinated, I would say, by butterflies. So they would pollinate while flapping its wings. Although it was a very strong trend, but yeah, it was not a significant work, but it's still a very good observation and something worth exploring in the region. And at least the study, the species which I've studied in the Himalaya, I never came across butterfly pollinators. So this was quite unique for the region. But I'm not very sure in the Himalayas because we have so much of so much of diversity of rhododendron. But still, till now, I have not come across any butterfly pollinators. Nectar volume. Nectar volume was explained significantly by birds. Here is the image from the Himalayas, which is Rhododendron arboreum and pollinated by Rufus ventationa. And arboreum had actually the highest amount of nectar volume. Yeah, there are definitely more bird pollinators in the Himalayas, starting from the sunbird and hoary-throated parwing. And it's true that warm-colored rhododendrons are preferred by birds. But I also observed in the field that bird do go for the other colors, especially when the abundance of warm colored flowers is less, they would opt for the other flowers with the similar rewards, and they don't care even if the reward is less. So having field observation is for these kind of small information makes it very important that we make these kind of field observations. And also I would say that just the color of the species sometimes would be tricky to rely on the pollinator group. So moving on, I also wanted to look at the role of the climatic conditions associated with the floral traits and the pollinators' identity. Firstly, so because the climate is very different for both these regions, what I did was I analyzed the similar data and saw that but I included climate and then looked at the interactions. So how I involved climate here is I took the occurrence data for all of these species and then extracted the climatic variables and did phylogenetic PCA, and my first two axis becomes my climatic variables. So which was majorly explained by temperature seasonality for Indian Himalaya region. And here again, even while looking at the interaction, I saw the nectar volume was explained by bird, and plus the interaction of bird with temperature seasonality, what concentration was negatively again explained by bird, whereas I did not find any trend in terms of corolla length. In terms of Appalachians, both nectar volume and concentration was explained by bumblebees, and although not significant, but I did see the very positive trend of concentration with butterflies and mean annual temperature with interaction with butterflies. So usually the butterflies are ectothermic and they usually need high temperature for basking. So, and what I observed is bumblebees were in the Appalachians are more active slightly in the higher elevation, while butterflies are more in the open and highly lit area. Yeah, that's why I think it would make more sense. Okay, so how I conclude that the common pollinators for Himalaya are birds, followed by bumblebees and flies. In the Appalachians is bumblebee, and definitely butterflies are very unique to the region. Bird-visited flowers with a higher volume of dilute nectar, while bumblebees preferred flowers with concentrated nectar. High nectar concentration observed in Appalachians and high volume among Himalayan species. Significant relationship between the floral traits, pollinators, and temporal seasonality were observed only among Himalayan species. Now, in terms of bird, I also observed bird visits in the Appalachians. These are, so you have only one single bird, which is a migratory ruby-throated hummingbird, but they would make occasional visits to the native North American species. Similar to what usually the butterfly would prefer, lower elevation species, but hardly would they interact with either stigma or the stamen. So they would not. They were not the common pollinators, I would say, but they did sometimes transfer the pollen, but not like how the bumblebees and the butterflies would do. And there were another form of bees. Yeah, another group of bees apart from the bumblebees. Again, the activity would be such that they would be more of the pollen robbers, but hardly would efficiently pollinate. So I would still consider bumblebee and butterflies as some of the important pollinator for the Appalachian region. So this work is still at the initial level. I started again when I was in the US, but it's still not yet completed. But I'm glad I did this because this helped me to work on my project at present. So what I'm looking for. So here I try to look at, are the floral traits in rhododendron affected by pollinators or a climate at the macroevolutionary scale? So the traits I looked at, which is corolla length, is from the published floras, published informations and the floras, and global occurrence from GBA fieldwork. Climatic variables, again, world climb, conducted phylogenetic PCA, use the 2-axis as the climatic variables. And I used for this the recent most resolved phylogeny for rhododendrons, which consist of 200 species, and used phylogenetic comparative methods. And selected the models based on the AIC values. So since in the regional level, I did not see trend on the any trend with regard to the corolla length. So I started with the corolla length and saw that it does show strong phylogenetic signal, meaning the closely related species show similar corolla length. But when I tried to look at the character evolution of corolla length for the entire tree, it did not make any sense. And what I did was then I ran the analysis for different clades, and what I found was estimated variance in rates of corolla length evolution varied across different branches. And it was interesting to see that a higher variance was observed in subgenus Rhododendron. Now, this is also the, it consists, at least in this phylogeny, it consisted of the species which are mostly in the Southeast Asia. Similarly, I used the climatic variables, and the PC1 was best explained by temperature seasonality. And here, when I looked at the global level, precipitation seasonality again seemed to be important variable. And again, for this, it showed that closely related species do prefer similar climatic niche. And again, similarly, when I looked at the niche evolution across the entire considering the entire tree, I did not see trend, but when I did the analysis based on different clades, the trend became more clear. Estimated variance in the rates of climatic niche evolution again varied across branches, and higher variance was again observed in subgenus Rhododendron. So there's definitely something going on in this particular clade, but in general, I would conclude that some clades are evolving faster or more variably than others in response to climate and ecological innovations. Although I've just started working on other traits, but yeah, some traits could explain very strong trend. Specifically, the corolla length showed higher variance in the subgenus Rhododendron clade. So maybe here there is more interaction between biotic and abiotic factors which needs to be tested, or it could be also explained by the biogeographic range. Now, when I saw this initial trend, that's where I applied for, where I thought of considering this topic as my Marie Curie postdoctoral research. And now I'm working at a Naturalis Biodiversity Center, especially on subgenus Rhododendron. Idea would be to construct the biogeography history and understand which factors can explain the shifts in diversification rate in this unique subgenus. And for this, I'll be collaborating with the Royal Botanical Garden, Edinburgh, and Kew. So overall, from all these findings, what I would conclude is rhododendron is definitely an excellent model to study the evolution of plant-pollinator interaction, and it does support pollination syndrome hypothesis to a large extent. And in the context of climate change, this result suggests that climate may potentially affect relevant traits such as the nectar, which has also been studied in various other geographical regions. And nectar, in fact, has been seen to be very sensitive to the rise in temperature. So we could definitely make use of this trait to understand the role of climate. And again, the phenology even seemed to be, the early even seemed to be highly sensitive to the climate. I use this rhododendron arboreum tree just to give you an idea of how tall the arboreum can grow in the lower belt. Yeah, and also it looks amazingly beautiful. Again, the nectar traits may be more reliable than floral morphology for studying the impact of climate and predicting potential pollinators. So every time I started my analysis and thought of incorporating evolutionary history, I had to give away some of the species just because the existing phylogeny did not have those species included. So I always felt that a resolved phylogeny with a very strong sampling fraction across the geographical regions is required to make more informed prediction about the evolution of the genus Rhododendron. Now, there are a lot of people to thank. Firstly, Subhadra, with whom I've worked in India. So I did my PhD from A3, and she mentored me. She was my PhD guide in Anahi. She was my postdoctoral supervisor in University of Maryland, and now in Naturalis, I'll be working with Renske and Lewis. So I've worked in tough terrains in the Himalayas, and I don't think without my field assistant support, I would have been able to sustain for long. So Nam was always there with my field expeditions, followed by Rakesh and Surya, who helped me to do my recent field work in Lachin, and Julia, who helped me with the sampling collection in Appalachians. Furthermore, I would like to thank Robbie and Shiv Prakash, who are my collaborators. And Robbie also worked in China on Chinese rhododendron phenology. And now Alan, who is helping me with their present work in RPG. And Juliana, who has always supported me with the required funding at the most difficult times. So I'm very thankful that I had this opportunity to work with all these amazing researchers. So thank you so much for listening to my talk, and I'll be happy to address some of your questions.
Wonderful talk. Thank you. Everybody ask your question by raising your hand or by putting a typed question in the Q&A. While people are thinking of their question, I'm going to start with what's a silly question. So there seems to be a popular idea in the US growing that hummingbirds pollinate red and orange rhododendrons. So I'm going to the source. I think you're the only person in the world who has data on this. Did you ever see a hummingbird? Is there any reason to think hummingbirds are visiting the azaleas in North America?
So maybe they mistake it to be pollinators because they are good visitors. They do visit. And I did observe in the field, but being a pollination biologist, we would always look for the transfer of pollen. So if the bird pollinates, then it would have a lot of pollen on their forehead and the neck. So when it bends, either the forehead touches the stigma or the neck has or touches the anthers. So although there were a lot of visits made by these ruby-throated hummingbird, but it did not transfer pollen. So it was a very occasional incidents where it did transfer pollen, but and it reflects in my data if you read my paper. But I would not say that these are most important pollinators, like how the pollination is facilitated by the butterflies and the bumblebees.
So what you're saying is they might be nectar robbers.
Exactly. Exactly. They come, and since they're so small and they have a very slender beak, they can easily get in the flower. And if you see, all of these azaleas have a very curved anther and extended and curved, so it can easily accept as it goes below the flower and just draw the nectar. So the 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 activity is such that it doesn't contribute much in the pollen transfer. At least that's what I've observed. But again, I would like to highlight that I just made observation for 7 species. In fact, I did more than that, but I had a big problem in identification of species. And ultimately, I got to understand that I did collect some colomorphs and I had to give away, but around nine different taxa when I did not see the contribution of hummingbird. It would be mostly like a pollen robber. Sorry, nectar.
Nectar robber. Very
Exactly.
fun. Thank you. Okay, we got 2 questions. First, I'm going to ask, Melissa had her hand raised first. So Melissa, why don't you go ahead and ask your question?
Let's see. For the macroevolutionary study, which flower traits were you looking at, and what is the geographic range?
It was corolla length. Geographical range for what?
Like which regions of the globe? Is it a full global look? Is it
Yes. So
okay?
it included all the regions, but then again, it was 200 species, and the geographical representation was, again, I would say, very less. But still, in the subgenus level, I would see a lot of variation, especially for the subgenus Rhododendron.
And then are you thinking about incorporating phenology into that study also?
Um, n- it's just 2 years project, so I'm not sure. But it would be very interesting to look at it. But I would not, having worked in the field, I would not trust the flora phenology information much, because it just gives you a broad range. And I had this experience before. Even if you average or take the first and the last phenology timing, it would be very difficult to see the trend. So I don't know. The answer is I'm not very sure.
Okay. I'm still trying to make sense of some of the tropical phenology stuff that I have put together.
Okay, nice.
So that's what I was curious. Um, what else was I going to ask about that? Oh, and then for let's see, the biogeography side of it, do you mean biogeography in the sense of the regions in which the species occur, or more getting into historical biogeography, or
Historical biogeography.
okay. Um, yeah, great. Well, thanks for your talk.
Thank you.
So we have in the Q&A, we have 2 comments from Tara Spears. She says, no question, just thanks for the presentation, and I look forward to reading your publications. As an employee at a botanic garden, we often get questions about pollinators and their relation to rhododendrons. This will help provide better explanations. Then we have one more question from Dylan. If phenology is genetically limited, how do you suspect the abiotic influences on plant pollinator relationships to be altered with climate change? Great talk.
Could you read that again?
If phenology is genetically limited, so I think referring to your phylogenetic similarity results of the early flowering stages, how do you suspect the abiotic influence on plant pollinator relationships to be altered with climate change?
So how I would see this is, so I was not able to present the entire data, but what we noticed, or what I infer from my phenology result is, since these early events are conserved, and even while incorporating the abiotic factor, it was still, we saw the strong role of evolution, a strong role of evolutionary history. So, in fact, if there is any rise in temperature, I would predict that all of these related species would behave in a similar way, and their interaction, or if there is any switch in the timing of or where when this species are exposed to the right temperature, if this is switched, just hypothetically in the future, then the way it would the way it'll get affected would be very similar to all of these related species. So I don't know if it makes sense, but I see that the closely related species are going to respond in a very similar way to the climate change. But interaction with the pollinator itself would be difficult to answer, because this also requires a lot of experimental work in the field, where we make use of the OTT, which is over top chambers, where we manipulate the temperature and see what is the visit like and what are the visitors like. And when we do this manipulation with the temperature, we see that how the species are responding to the rise in temperature and how it's interacting with the pollinator. So without those detailed information, it would still be very difficult to answer the question with regard to plant pollinator interaction in relation with the abiotic factors.